Basically, it's my pleasure to introduce Professor Nagarajan Vaidehi. She is the chair, Department of Computational and Quantitative Medicine, Beckman Research Institute. And I see that, it's great to know her also. And I see that she does a lot of work on protein-protein interactions and protein ligand interactions and drug designs using physics-based modeling, molecular dynamics simulations, and other techniques. So I would now hand over to Professor Vaidehi, and I think the talk will be for an hour, and then we will have discussions after that. So, Professor Vaidehi, maybe you can start.
Thank you. Can you all hear me? Can you hear
Yeah,
me?
we can hear you. We can hear you.
Okay. So, thank you, Arnab, and thank you, Prabal, for inviting me to give this talk. And I enjoyed some of the talks yesterday. I tried to join later in the evening, the first and the second talks in the morning. So I really enjoy the variety of talks that you have put together. It sounds like a great conference. Yeah. And I was looking forward to being there in person, but given the COVID, I don't mind doing this because we have to continue sharing science with each other. So with that said, I just want to introduce you briefly to what City of Hope is. So City of Hope Cancer Center, it's a National Institute of Health designated cancer center. We have a hospital where we do clinical research, and we have the Beckman Research Institute where we do basic sciences. The idea here is to be able to translate the basic sciences to the patient. So we have the capability to develop drugs as well as to synthesize it with the FDA quality and then and try it on patients and conduct clinical trials all in the same campus. That's what is exciting as a computational chemist. That is a very exciting thing to us. So at my department, the approach we try to take is like a Mars, Mars rover type of mission. So in universities, typically people work in smaller groups, and they are doing basic sciences, and that is a very important critical part. But in here, we try to form a disease team. So, for example, we believe that if we can, land a rover in Mars precisely at a prescribed location, millions of miles away, we should be able to land a drug on tumor with precision. And this precision is so important because that will avoid side effects of the drug. And then cancer, I think more than the disease itself, the treatment is also dreadful. So we want to minimize that as much as possible. So with this decision, we form what's known as a space mission type of approach. So we have different disease-oriented teams, right? Like a pancreatic cancer, we'll have a clinician who's doing surgery and treatment in pancreatic cancer and doing research at the clinical level. We have molecular biologists, and we have basic scientists like me, computational people, and also people who do organic chemistry. So we form these teams and meet, and my group is part of this team. And we want to form this kind of space mission. So everybody's goal is to be able to solve this problem as much as we can, while we go and also do our own research, basic research, to build tools that will solve the problem. So if you were to look at simulation, this Prabal will be very familiar. We both learned this at Caltech, that if you were to look at all the processes and a time scale that happens in biology, start with a human, and you have a length scale in here, you have these different organs, right? We want to be able to understand what are the processes that happen in an organ at a cellular level that can bring about change and disease condition, and can we predict prior to going to the disease condition. And so there are people who are doing simulations at an organ level. Can you simulate the actual workings of the lung, right? So they do. In my department, there are faculty who deal with that. So at very length scale, high length scale and high time scale. And we also have faculty who look at the tissue. So if you go down into an organ, take a piece of the tissue that makes up the organ, how do these cells interact with each other? So if you are given the content of the cell, for example, the genomic, the RNA sequence content, and as well as different protein levels in the cell that makes up the cell. And if you are able to give this information as a function of time, can you simulate how this tissue behaves or cell to cell communication happens? And therefore you can simulate how a tumor grows. And then in the presence of a drug, you can simulate how the tumor actually responds to the drug or not. The idea is that if we can simulate the cell to cell growth, we can see how much a tumor grows, or we can match it to the MRI of the patient and predict if the patient is going to respond to a particular drug or not early on during the course of the treatment, rather than waiting for a longer time to measure the response. And then if you zoom into the cell, there are processes that happen in milliseconds, and you know how different proteins go from transport from one particular organelle of the cell to another organelle, and what is the cue that gives them to, or makes them move from one, so you understand the dynamic motion of proteins from different organelles depending on the signaling pathway. And you zoom into these proteins, and then you see protein complexes are actually working together at microsecond and hundreds of microsecond time scale and micrometers length scale. And you take one zoom into further one of these proteins, you want to understand the workings in terms of atoms, like, you know, how do the atoms come together in this protein and change the conformation of this protein. So this is at a nanoscale and nanosecond time scale. So we have faculty working at all these levels in my department, and we try to interact and see what are the techniques we can use that forms a common thread. And I'll give you some examples as we go along today. So my lab in particular has been focused on one part of this chain that I'm showing. So in the 60s and 70s, when they sequenced amino acids, when they sequenced proteins, and they tried to look at similarities in sequences, amino acid content of the protein sequences, and match it to the function of that protein. If two proteins, for example, one of it is a kinase and another protein is highly conserved or highly homologous to the other protein, then we believe that, they believe that it could be a kinase as well. So predicting function based on sequence similarities. In the 80s, when the structures, three-dimensional structures of the protein started coming out, and then people compared different folds, like the three-dimensional fold of proteins, and then correlating that fold to function. For example, antibodies, and there are other proteins which are very diverse in sequence, but in the sequence space, but they have a common structural homology, and they could be related to function. But now we realize that more and more of the proteins that are in the human cell have intrinsically disordered region. And also the structure is not just one static snapshot, right? We want to be able to understand how, when you put the structure, when they actually solve the structure, they crystallize the purified protein. So it's in a solid state. When you actually put that protein structure in the real cell condition, how does the protein structure change in response to the cellular conditions? And how can we correlate to the function? And more important, there is no structural information on proteins which are proteins which have regions that are disordered, and some proteins are completely disordered, but they are also important function. They carry out important function. So in order to understand the role of intrinsically disordered regions in proteins, we also need dynamics. So my lab has been focused on how to delineate dynamics of protein structures and how do you correlate it to function. So we typically use the classical molecular dynamics simulations, a different variety of it, and I'll show you. And all our projects are always in close collaboration with experiment, because we really need to have balances and checks as we make predictions using these simulation techniques. So the topics I'm going to cover today are I'll give you a brief introduction of multi-scale MD simulations and what my lab has contributed to this. It's a very big field. There are many other labs which are also working on this. And I'll try to give you one example of how we can use this multi-scale MD simulations in order to understand the role of intrinsically disordered region. And I'll give you examples in kinases. I will talk about how protein-protein interactions, dynamics plays an important role in protein-protein interaction, and how do you use dynamics to understand structural determinants, and also allosteric communication. I will talk a little bit about it, and I'll tell you what it is as we go along. So the first part I'll be covering is the multi-scale simulation. So when you do, so typically in molecular dynamics simulation, the all-atom molecular dynamics simulation, you solve the Newtonian equations of motion as a function of time. You calculate the forces using a parametric force field function. And the parameters for the different forces are coming from quantum mechanics. And we solve the Newtonian equation to get the trajectory of this system. So as a function of time. But what happens is if you take a crystal structure, let us say that protein undergoes conformational changes, major conformational changes. This is one functional structure of the protein, and this is another functional structure of the protein. When you do all atom dynamics, it typically stays in this conformation depending upon the barrier. If the barrier is lower, then it would go transition and sample all these states. But if the barrier is higher, it's a different functional state of the protein. It's harder to. So people have enhanced sampling methods. There are multiple flavors of enhanced sampling methods. I'm going to talk about two particular ones. One where my lab has been involved in very intricately. So in the Cartesian Newtonian dynamics, what we do is we track the trajectory of every atom moving in the field of all the other atoms, right? So you have forces from bonded atoms, you have forces non-bonded atoms like Coulombic and van der Waals forces and so on. So you're tracking every atom. But if you want to look at the overall conformational, large-scale conformational changes in proteins going from one state to another, you can actually freeze the high-frequency degrees of freedom, such as bond length, bond vibrations, and also bond angle bending. These frequencies, these degrees of freedom may not contribute substantially to changes in the dihedral angles. So changes in the torsional angles between these amino acids, both backbone and side chain, contributes to this large-scale conformational changes because they are the low frequency degrees of freedom. So what we do in our coarse graining is model this whole protein like a cycle chain. For example, these are rigid bodies connected by hinges. The hinges are the torsional degrees of freedom. You can do this for a large protein. You can have multi-domain. I'll show you in the next slide how we do this on a larger scale coarse grain. The idea is to be able to sample, go over the barriers in the torsional degrees of freedom and sample multiple states. And then the other way to do coarsening is what people do is if you take this all atom model of a molecule, instead of considering all the atoms moving individually, you can cluster, a bunch of 4 carbon atoms as one larger sphere. So it's called a coarse grained particle. So benzene is 1 sphere or 3 spheres, and then 4 carbons is 1 sphere. So this is coarsening the force field. In here, you coarsen only the dynamic model. The forces are still calculated for all the atoms explicitly, so you don't lose the granularity of the hydrogen bonds and so on. In here, you are coarsening the force, so the particles as one big particle. So it's more of an approximation, and I'll show you what kind of information you can get from coarsening this or coarsening this way. So this is coarsening the force field, this is coarsening the dynamic degrees of freedom. Ultimately, if you can sample these different conformations, you pull out conformations from these 2 different potential wells, and you do all atom simulations and calculate accurate thermodynamic properties from that. So this is the approach we have taken. So this the approach that I said about freezing the high frequency degrees of freedom and performing the molecular dynamics simulation in the torsion or internal coordinates is called the Newton-Euler inverse mass operator method. The problem here is that the equations of motion, becomes coupled. That is, they are in the dihedral angle space, so they are coupled. But so that was intense, like computationally intensive. It scales as order n cubed, n being the number of degrees of freedom. But we borrowed these algorithms from robotics and worked with them to adapt their order n algorithm, from a whole field of mathematics called spatial operator algebra. And then that scales linearly with the degrees of freedom. So by doing this, we were able to traverse through the potential energy surface. So you're essentially sampling your protein conformational changes in response to the forces around it, only in the torsional degrees of freedom. So you're giving the velocity to the torsions, and therefore you're able to sample the conformational changes. So we did several applications on this, and we did like folding of very small proteins, about 80 amino acids. And then we did structure refinement of proteins, that is, take homology models of proteins that are approximate and try to refine them and see whether you get close to the crystal. And we took part in this competition called CAS. And then we also did quite a bit of thermodynamic property restructuring in the internal coordinates, right? And we developed a code for it. And we also studied domain motions and so on. So I'll give you one simple example of what we can do practically for studying intrinsically disordered proteins in here, right? So so here is a kinase. So the classically cancer drugs are typically one of the largest used cancer drugs are kinase inhibitors, right? So kinases, and many kinases actually, it's a large family of proteins, and they are a multi-domain protein. So they have a catalytic domain where a substrate protein binds, a part of the substrate protein binds, and then there's ATP binding site in the other domain. So this is the catalytic domain of the kinase. And then the phosphate group, it's transferred to a serine or threonine or tyrosine on the substrate. And then this substrate leaves, it gets phosphorylated. So kinases phosphorylate other proteins. They can bind to multiple substrates and then transfer the phosphoryl group. But so when inhibitors were designed by pharmaceutical companies, they targeted this ATP binding site. Typically, they are ATP competitive binders, the inhibitors. So they bind in here, they do not allow ATP to bind, so this preventing phosphoryl transfer, and therefore the catalysis of this kinase catalytic domain is inhibited, right? But this ATP binding site is highly conserved across this large family of kinases. So you're going to get cross-reactivity of these inhibitors across kinases, which will cause side effects. But we neglect this effect of all these regulatory domains. So this particular kinase is called PKC, belongs to the AGC kinases. And
sorry to disrupt, there is a question in the chat box. Arnab, you might want to pick it up.
I'm sorry?
No, I'm telling Arnab, there's a question.
Yeah, I thought that, should I do it at the end of the talk or should I do in the middle? I was not sure. So Professor Vaidehi, are you okay asking questions in the middle of the talk?
Absolutely. We can cover that way the 1.5 hours. If you want to interrupt with question, that's fine with me.
Sure. Okay. So there is a question in the chat box. There is asking that what exactly does intrinsically disorder mean? Does it mean protein keep changing its structure?
Okay, so intrinsically disordered means when you take this, I'll explain as I explain this case also, okay? So this protein kinase has multiple regulatory domains. This is the whole polypeptide chain of this kinase. These domains actually interact with this catalytic domain and keep that in a resting state. And some other, like calcium or other molecules, bind to these regulatory domain, and then they open up the catalytic domain, right? And these polypeptides that are between these regulatory domain are linkers. Okay, so those are linkers, and the crystal structures have solved the catalytic domain and the regulatory domain separately, but they couldn't solve the structures of linkers because they are disordered. So what I mean by disordered is they do not have any secondary structure elements in here. There are no helical structures, or so they do not form a three-dimensional ordered fold in these regions. These the linker 1, V1, V2, V3, V4, and V5. These four regions are all disordered in the sense they do not have a typical fold. But what happens is they may have an ensemble of fold, and when the C1a, for example, interacts with this catalytic domain, like it comes around and interacts with this catalytic domain, and then that particular linker may take a certain fold. Okay, so I'll show you in the next thing. For example, this is the catalytic domain, and this is the domain C1a, okay? And this is the linker, disordered linker shown in blue. So it doesn't have any, it has one turn of the helix according to our prediction, and it doesn't have any order at all. So any order in the sense, it may take an ensemble of conformation depending on the dynamic conditions around the protein, but it doesn't have as one stipulated fold. For example, this is a typical fold of the catalytic domain. This is the typical three-dimensional fold of the regulatory domain, but these linkers don't have any fold. So what we did was we treated with this Newton-Euler, the GMEMO method, the internal coordinate dynamics method, we treated this whole thing as a rigid body, and then this linker as flexible torsion, and this is another rigid body. But we allow the side chains to move, only the backbones are held rigid. So then what happens is, I'm not showing the solvent in here just for clarity. We can traverse that, take this disordered link region and sample all the possible conformations to see whether the C1a, where does it interact with the catalytic domain, right? So now we found this is one possible interaction site. So we derived 200 different possible ways C1a interacts with this catalytic domain, the regulatory domain. And we took the most energetically favorable conformations of this domain with the catalytic domain. And what we identified, we have shown top scoring 5 models in terms of the energy. The red one is the most favorable, but this magenta one is least favorable. When I say least favorable, they're all very similar in energy, like 0 to 70 kilocalories interaction in the interface. So you can't really tell in this coarse grain level that these are very different, right? So you have to test out all these models experimentally to see which one of these model regulatory interaction with the catalytic domain is actually likely to happen. So we have these, yeah.
Yeah, so I have a question here. So, sure. So you are using internal coordinate dynamics compared to traditional what people do as Cartesian dynamics. Mm-hmm. So could you tell us that what is the advantages in this particular method to find out these different this thing in an overall manner?
Sure. Yeah. So the way I think of it, I can give you an analogy, like, you think of Grand Canyon, right? So you want to, the bottom of the canyon is the Colorado River. So if you want to raft on the Colorado River, the Cartesian dynamics, what it does is, it does the bond vibration. You wasting all the velocity, some of the velocity, of course, it's a stiff degree of freedom, but bond angle, right? You give velocities to it. It's like going up and down the canyon as you are rafting down. But if you only raft down and not go up and down the walls of the canyon, then you're following a path of minimum energy. So what I'm trying to say is that you're giving in these dynamics all the velocity to only the torsional degrees of freedom, okay, right? And therefore you are able to sample torsional degrees of freedom collectively give rise to larger scale transitions between conformational transitions in proteins, right?
But
Because
that may have some issues with let's say when you are doing torsional dynamics. Don't you face problems in getting trapped in local minimas based on the interactions from the side chains?
yes, so but we do, I mean, in the case of in the case of Cartesian, it's even more, right? So that is what I'm trying to say is you can get trapped in local minima when you're tracking bond vibrations, bond angles. They may get also trapped in, for example, if you were to look at domain motions that they have, in the previous papers, what we have shown is the domain motions that were observed in small proteins in Cartesian dynamics, which were 400 nanoseconds, we could map that within 50 nanoseconds in GNeMo. So what I'm saying is it is highly likely it can also get trapped. But since you're giving a lot of velocity to only the torsional degrees of freedom, you are able to you're able to surf over the conformational space. But I want to talk to you a little bit about the caveats of this. The torsional, once you make the bond angles and bond lengths stiff, what can happen is, so if you especially if you stiffen the bond angle degree of freedom, bond angle the frequency of bond angle is not exactly in the region of torsion, but they overlap. So the changes in the bond angle assist dihedral transitions. So what we found was that the backbone bonds, freezing up the backbone bond angles in proteins is not facilitating the conformational transitions in the dihedral. So the barriers for dihedral transition in this dynamic model is higher than in Cartesian. So what we did in this internal coordinate molecular dynamics is to open up the bond angles. So in this model, what we can do as a user is to say, hey, can you open up to the model? In the model, you say freeze all the bond lengths because they are very stiff and harmonic and high frequency, right? And then freeze only the side chain bond angles, but not the backbone bond angles. Keep them open. So it can do mixed internal coordinate dynamics as well. So that works much better in not posing a stiff model.
Okay, thank you.
Sure. So these top 5 models, what we did was we looked at and we did all atom MD Cartesian dynamics in all of them to see if this domains actually stay together when you do all atom dynamics, Cartesian, which is more accurate thermodynamically. And what we did was to map the interface between the regulatory domain, which are the residues in the regulatory domain that's interacting with the catalytic domain, and we mutate each one of these interface residues to 19 other amino acids and calculate the change in the mutation, and the change in the interface energy upon mutation for all these 5 models, because we don't know which one of these 5 really is going to pan out, right? And then we predict mutations. And what we found was when we did the mutations and did the predictions, we found only model 1 and model 5 showing the best change for mutations in predictions. So model 1 and model 5 gave best energy in terms of all atom interaction energy. So this is the coarse grain, from the internal coordinate molecular dynamics energy, but the all atom energy showed these two as distinctly different from the other, as well as the mutations that were predicted showing a big change. So we predicted mutations that will increase the activity of the kinase as well as decrease the activity of the kinase. And those were tested by experiments by our collaborator. And they found that the model 5 and the model 5 gave based on the mutations that we predicted, gave the best change in the energy, in the activity that we made the prediction. So we think that the regulatory interactions these are the amino acids that are involved in this protein-protein interactions. See, it's very hard to solve this crystallographically only because the linker is disordered, and also these interactions are transient. In the cell, they don't happen and stay that way. Only when calcium binds to this domain, it interacts with the catalytic domain and it goes away. So these are weak, dynamic, transient interactions which cannot be probed into using NMR or crystallography or any of the EM, any of the conventional structural biology models. So now what's the use of knowing this? What we want to do is, this is the ATP binding site. We want to be able to identify a small molecule binding site that is less conserved across all kinases, but that will have an effect on the ATP binding. So it's an allosteric site that has communication to the ATP binding, and therefore we can target that site. So this is the interface that we map using mutation experiments and our multi-scale MD. And we identified putative small molecule binding sites. So we searched this whole surface across the dynamic snapshots and looked for any cryptic binding pockets, small molecule binding pockets, and identified small molecules that could bind. And they are testing these small molecules here. And so hopefully we'll find an allosteric modulator. So the caveat is we can find good binding molecules and they will affect ATP binding, but we don't know whether they're going to enhance ATP binding or decrease. So that is something we cannot tell from the theory part yet. So whether this allosteric modulator will be a positive modulator, which enhances the activity of the ATP, or it's a negative modulator, which decreases the activity of ATP and therefore acts as an inhibitor. So, but if we can find small molecules to this area, the amino acids here are less conserved. So you will get a kinase specific inhibitor, which is a good thing. So the next two topics that I'll cover is, now I talked about the intrinsically disordered region, but I just want to talk about even the ordered structured region when two proteins come together, how dynamics plays an important part in understanding which amino acids in the interface are important for this interaction. And also I'll talk about allosteric communication in proteins. So for this, we use the coarse grain model where we coarsen the atoms, the coarse grain force field, because I'm interested in membrane proteins for looking at protein-protein interaction, and membrane proteins are present in the cell membrane. See, we want to simulate the actual the actual forces of how the other lipids. We know that lipids play an active part in modulating the conformations of the membrane proteins. So if we do not put in the lipids, just like mimicking a cell membrane protein, we may not get the exact biology of it captured in our simulation. So we try to do, I'm not saying this is the exact way it is in cell membrane, but we do put in multiple different lipids that are known to be components of the cell membrane in the outer leaflet, of the cell membrane bilayer, and the inner leaflet in this ratio. And we try to, the proteins are shown in mustard, and then only some of the cell membrane components, for example, PIP3, we wanted to see how PIP3 interacts with these proteins, are shown in green. So how do we use this coarse grain? So the coarse grain, as I showed, 4 carbon atoms become a sphere, right? So it's a very crude model. It is useful only for looking at aggregate or kind of a gross property. If you want to check whether a particular protein in the membrane dimerizes or oligomerizes, it's a good force field. If you want to look at how lipid rafts are formed, like, in the cell membrane, there are compartments that are formed in which the membrane proteins sit and signal. So those are called lipid rafts. If you want to understand how lipid rafts are formed, which lipid components form the lipid rafts and so on, like you could see the PIP3 was going towards the protein, right? So how they aggregate and stiffen the protein there, all this kind of properties, more gross properties, can be understood by this coarse grain. But what we do is we create like 5 such magnified samples, because only the ratios there, how they should be organized in cell membrane, we don't know, right? So we start with 5 different random conformations, and equilibrate using this dynamics, using this coarse grain dynamics over, we perform this coarse grain dynamics over several microseconds. And then we pluck out parts of this parts of this coarse grain, and then you convert it to all atom, and then you perform the simulations. Then you know exactly how lipids at an atomic level interact with the protein. With that, I'm going to go to application now. So G protein coupled receptors form these largest class of drug targets. They are superfamily of drug targets. They are membrane proteins. They have 7 helices going across the membrane from outside the cell to inside the cell. They, when ligands bind from outside the cell, right, the ligands can be as small as a photon, or the ligands can be peptides or small molecules, or large proteins can also be ligands to these GPCRs, this class of receptors. And upon ligand binding, about 3 nanometers away, 30 angstroms away, when a ligand binds in here, this protein opens up in the intracellular part, and it couples to a family of proteins called the G proteins, guanidine binding proteins. And that's why they're called G protein coupled receptors. And once it couples to a particular G protein, if it couples to this G protein, it activates adenyl cyclase, and that leads to a biological effect in the cell. So it's one signaling pathway. If the same G protein couples to another same GPCR, when a different ligand binds, it couples to a different G protein, and it leads to a different signaling pathway. So single GPCR can couple to multiple G proteins depending on which ligand binds to it. How does this ligand protein pair, ligand GPCR pair, couple to selectively or even promiscuously to different G proteins is what we want to understand. What is the atomic level mechanism by which information from outside the cell is passed on, transduced to inside the cell, that makes this coupling either selective or promiscuous? So now, large scale experiments. So what they do is people express this GPCR with one of the fluorescent proteins, this G protein with another fluorescent protein. And when they add different ligands, they tested this on 150 different GPCRs and they calculate how well does given GPCR couple to different family of different members of the G protein family, right? So for every GPCR, they will have a whole set of G protein coupling efficiency. And what they found was certain GPCRs, I mean, you don't have to see what's written in here, but they're all GPCRs. So certain GPCRs are very selective. That is, when it ligand binds in there, they couple only to GQ and not GS, for example. GS and GQ are 2 different G protein families. But some of the GPCRs are promiscuous. They couple to GQ or GS and with equal efficiency, so coupling efficiency. How does this come about is what we wanted to understand. If you look at the, I just went through this quickly. We wanted to see whether there is some kind of evolutionary pressure to go from selective to promiscuous coupling. So we took one subfamily of GPCRs, the adrenergic receptors. There are 9 members of adrenergic receptors. All of them bind to adrenaline and couple. And this is the 4 different G proteins that they couple to. If you see a black sphere, that means they couple to that particular G protein. If you see hatched one, then they don't couple that efficiently. White means they don't couple at all using these experiments, the fluorescence resonance energy transfer experiments, FRET experiments. What they found is these members of this adrenergic family, some of them are promiscuous and some of them are very selective. See, only one circle is filled, the rest are all. That means it's selectively couples to only one of the G protein family members, not the other members in the family. And some of them are promiscuous. So, but when you look at the evolutionary tree of this subfamily, these have come later. So did they go from being promiscuous to selective? But we said, okay, we'll look at another subfamily, adenosine receptors. They went from selective to promiscuous. And we looked at opioid receptors. opioid receptors, they don't show any direction in their evolution. So there is really no precursor that all GPCRs were originally promiscuous and they become selective. We couldn't say this from this. So they are all, every GPCR exhibits a spectrum of coupling specificities. Now, what we wanted to know is, what are the amino acids? So this is a GPCR, and this is the G protein, and this is the crystal structure of the GPCR G protein that gave Brian Kobilka the Nobel Prize in 2012. So if you look at all the interactions in this interface in here, in the crystal structure, can you tell why this particular GPCR couples only to this G protein and using that structural determinant, right? So we looked at the part that actually is in contact with the GPCR, which is this helix of the G protein is a big protein, but this is the part that interacts with the GPCR largely. And this is GES protein. If you look at another structure, crystal structure, for another GPCR, also with the GS protein, you see the slightly different orientation of the same helix. So these two are the same protein, but they bind in different orientation. So if you look at family of GI, more structures came out, you see that they are all different. I mean, they are in slightly different orientation. So what we think is there is heterogeneity in these conformational states. The coupling is not one state, it's an ensemble of states that is exhibited even by just looking at the crystal structures. So this interface should be dynamic. And what people tried to do after the crystal structure.
I have a question, Prabal here.
Yeah, sure, Prabal.
Can you go back to this previous one?
Yeah.
Yeah, so these are like on the left, these three different structures, correct?
The GPCR is different. There's cannabinoid, opioid, and rhodopsin, but the G protein is the same, identical.
But this different conformation, this is from the crystal structure?
Yes, this is from the EM, cryo-EM structure.
Cryo-EM structure. So could it be that in the cryo-EM, so this is in the cryo-EM also, they just give you a series of structure, but this is one of them only. In cryo-EM also, they might have like in same structure, they might have got all three.
Right.
They
but
don't,
this was, this was solved to 3.5 angstrom, the cryo-EM structure. So they did not find alternate conformations. But you're right, in another GPCR structure with a different protein bound called arrestin, they did find two conformations, even in the single cryo-EM structure, like you said. But these were all, they found only one orientation. But this orientation, what I'm trying to say is it's very flexible. I don't think this is single orientation. In the cryo-EM condition, or the condition in which they purified these proteins, under that buffer condition, they found this orientation. So if you look at the GPCR G protein contacts just from this structure, it will not explain why it is selective to GI. So that's what I'm going to show in the next slide. Does that answer your questions, Prabal?
Yes.
Okay. So if you take all these structures, and then the shown by the lines are the GPCR G protein interacting residues, right? So now what people did was to, this is Madan Babu's group at Cambridge. What they did was to look at the sequences of the G protein, or the residues that make the contact in the G protein that make contact with the GPCR. Are they conserved? And they looked at all the residues that make this contact interface, and they found these are conserved. So they call this particular residue shown in pink as the barcode. That means these residues are the ones that are recognized by the GPCR. But they could never find in the GPCR a barcode like this, because GPCRs are by themselves a lot more diverse, not just than the G protein. There are only 5-6 different types of G proteins, but there are 600 different types of GPCRs. I mean, different sequences of GPCRs. So they could not actually get a barcode on the GPCR side, but they got a barcode on the G protein side. So what we found was, if we look at these interactions, the GPCR G protein interactions, I'm showing a zoomed in view of one of the interactions. So this is an arginine in the intracellular loop of the GPCR. So that's the gray. And this is the G protein in here. So this arginine is interacting with the backbone of the leucine in the EM structure. But there's an aspartate nearby, there's another arginine. When you do the dynamics, this arginine is not sitting quiet. It is interacting with all these residues in its vicinity. So if you were to look at the crystal structure, you will think only the leucine is important, or the EM structure, sorry. Only the leucine is important, but actually this arginine making a salt bridge with this aspartate and interacting with this threonine. So all these residues become important. So what we did was to do dynamics on all these complexes. All these are coupled to GI protein, these are coupled to GS protein, and this is coupled to GQ protein, and this is also GI. So whatever EM structures were available, we simulated these, and we looked at all the interactions that are made across the molecular dynamic simulations in the cell membrane conditions. Yeah.
Professor Vaidehi, are they coarse grain simulations, or are they all atom?
So first we did the membrane in the coarse grain, and then we took the membrane model, 5 different models of the cell membrane. This is a complex membrane with different components, right? And then we put in this and did all atom simulations on these. Because these interactions in coarse grain will not be good. Because the coarse grain doesn't have the directionality of hydrogen bonds, right? So we have to do these in atomic scale.
You use the internal dynamics, internal motion dynamics?
No, here we used all atom. We did the coarse grain using Martini force field for the cell membrane, and we went to all atom dynamics straight, no internal, because we really want to get the atomic level properties here, right?
Okay. Thank you.
So that's why we did all atoms. So this is a different multi-scale protocol using coarse grain Martini force field followed by all atom dynamic.
Okay, thank you.
Is that clear?
Yeah.
Sure.
Yes.
So we calculated the heat map, what I call as heat map for these protein, GPCR G protein interaction. I'm just showing a small slice of it. We have hundreds of these interactions. What are these interactions? The interaction of protein A with protein B, in this case, the GPCR and the G protein for these different complexes, right? And you see that their frequencies are all different. So the frequency of interaction, how often are these interactions persistent in the MD simulation? We ran 5 microseconds for each complex. And then we tried to do multiple simulations, not just one, and then aggregate and calculate the heat map. So the idea is that you would think that that if there are interactions that are common among, I just want to go back to the previous one. If there are interactions that are common between GI and GS coupled receptors, these interactions are common in the sense the same residues are making contact, you would think that they would not contribute to selective coupling, right? But they will, because in this case, if it is only 40% persistent, and this case it's 90% persistent, this is going to contribute to this free energy of this interaction a lot more than this, right? Because of their population of that interaction. So we think that this frequency map is this is the spatiotemporal heat map of these interactions are very important. So we took this large 700 different interactions, and we did this linear discriminant analysis. We wanted to see which one of, so this is a G protein residue, and this is a GPCR residue. So these are pairs of residues. We want to know which one of these actually contribute to selective coupling to GI versus GS versus GQ. There are 3 different families of G proteins, right? So the GQ is here, the GS is here, and the GI. So we could find these interactions that are discriminative of these G different couplings, right? Selective coupling. So now we go and look at the weights of all these different interactions in component 1 that contribute to high weights to component 1 and the linear discriminant component 2. And we came up with what we call as the QR code. So that's a one-dimensional code from sequence analysis. From dynamics, we've come up with actually a three-dimensional code. The reason I call it three-dimensional is we have the GPCR residues, we have the G protein residues that contribute selectively to coupling only to GS, those residue pairs that couple to GI, and those residue pairs that couple to GQ. But we also see variation in their frequency. So this is a dark frequency, which means this interaction will contribute very strongly, but this interaction will not contribute so strong to GS coupling, right? So there are some, what we found was these selective coupling residues are all this, you're looking at the interface of the GPCR where the G protein interacts. Did someone ask a question?
So actually, I wanted to ask one question. So Arnav here. So spatiotemporal, when you say, I understand the frequency as you are doing a time average frequency, but how do you take care of the distances? So for example, is the contact 0-1 indicator based, or is it based on a continual changes?
So that's a very good question. So we have like a cutoff for the distance. If it's below 3.5, we call it a hydrogen bond yes 0. I mean 1. So it's 0 if it's not above 3.5. We are not looking at the rheostat kind of thing.
Yeah. That would be much more complicated actually. Yeah. Okay.
We are doing that for micro switches. I mean, I'm not going to talk about that today, but we are in the process of doing it. Okay. So what we found was, now can we test it experimentally, right? So that's we collaborate with Shiv. He's in the University of Minnesota, and what he does experimentally is to express this protein as a single polypeptide. So this is a GPCR. This is a donor fluorescent protein connected by a linker of stipulated size and length, and then there's an acceptor fluorescent protein, and at the end of it he puts the G protein in here, shown as this peptide. So this is the peptide that that inserts, I showed you in the previous slide, the helix that inserts into this. Sorry, I'm going way forward. This part. So this is the one he uses in the sensor. So yeah, sorry. Okay, so what he did with the sensor was, so when you treat this sensor, he expresses this as a single polypeptide chain, what he calls as a sensor in the cell, and he treats it with an agonist. And if this G protein were to couple with this GPCR, it will come here, and then the fluorescent intensity will be altered, right? So, and the change in the fluorescent intensity is what he measures. So now what he measured was, he took 6 different receptors. These receptors are known to couple to GS, and these are known to couple to GI, and this is known to couple to GQ. And what he found was, with this FRET sensor, he found that the S peptide couples to a shows very the GS coupled receptors show selectivity to S peptide, and the GI shows selectivity to the I peptide, GI peptide. So this is the no peptide, S peptide, I peptide, and Q peptide. This is the sensor. He changes the sensor with the different peptides in here. And he takes the Q receptors, and they also show selectivity to Q. But you see that this receptor is more promiscuous because it also shows, it couples to S and I and not just Q. So, and what we did was, we did this MD simulations. We calculated the interaction energy between the G peptide and the G protein and the GPCR, and match the interaction energy, the binding energy of this peptide. We calculated that and match it with the FRET. So on this scale is the FRET, and here is the binding affinity of the peptide to the GPCR. And that matches qualitatively to the FRET intensity changes. So now what we did was to look at the peptides, the 3 peptides in which we simulated, we calculated which residues in this peptide show a high interaction with the receptor, so high interaction energy and also sustained interaction. And these residues that are in these blocks are all high interaction, what we call as hotspots, selectivity hotspots. What is interesting is, if you were to do just sequence analysis of this G protein like Madan did at Cambridge, he would have predicted this conserved residue would never contribute to selectivity, right? But as I said, the spatial-temporal interactions, the frequency can be modulated. It's a hotspot in the GS. Although it's conserved in GI, it's not a hotspot. And it is, and the ones in bold are really hotspots. And in GQ, it is a hotspot. So we tried to predict hotspot and test it experimentally. So what we did was, can we take a GS coupled receptor and the S peptide and make Q-like mutation? And we showed that when we made Q-like mutation in this hotspots in the S peptide, the coupling reduces to the S receptor. But to the same S receptor, the Q peptide doesn't couple that well in the wild type. But when we make S-like mutation in the Q peptide, you see an increase in intensity. That means we took an S peptide, made mutations in here, and it started coupling and showing this state. So now, when we did this, we also did MD simulations simultaneously on the mutants. We find that the mutants actually reorient themselves from an uncoupled state to an inserted state in here when you make these single mutations in here, right? So, and we said, okay, with these single mutations, we did this for the Q receptor as well. You know, we took the Q peptide, made S-like mutations based on our predictions, and the S peptide Q-like mutations based on our predictions, and it showed decrease and increase for the Q peptide as we expected. Now, with these single mutations, do they also signal? These are just coupling with FRET, but do they also produce the secondary messenger and the actual biological effect? Yes, they do. So this is a Q-coupled receptor, but when we made single mutations in the S protein, GS protein, single mutation, it started coupling to the GS protein. This is a Q-coupled receptor. It started coupling to the GS protein with a single Q-like mutation, and then it produced these secondary messengers in cells. So now we asked, can we go complementary? We made these mutations in the G protein. Can we make complementary mutations in the receptor so you can make a selective receptor into a promiscuous receptor? So we predicted the mutations, 3 mutations in this receptor that would make this beta-2 adrenergic receptor, which is GS coupled, selective, and we made these mutations to make it the triple mutant, made it coupled to GQ. So what I'm trying to drive home is, by making and confirming with these experiments, we showed that this promiscuous receptor, beta-2, now which couples to both GS and GQ, we saw a rearrangement in the intracellular part of this protein-protein interface that there is. So this protein-protein interface is very plastic, right? It didn't couple to GQ, but it did not have the hotspots for it. But once we put in these mutations for hotspots, it coupled to both S, GS, as well as GQ in this orientation and GS in this orientation. So what I'm saying is, these things cannot be probed experimentally, the interaction frequency, because the time scale is very short, and NMR, none of those can probe these interactions in this transient interface between the GPCR and G protein, and we could do it. I think I'm running almost out of time, but I'll quickly go through how we have looked at the allosteric regulation as well. So when I said that when a ligand binding downs, we found out what are the interface residues that makes it selectively couple to G proteins, right? Different G proteins. But what are the residues that are involved in between? Can we map all the amino acids in between that mediate this communication, which is 30 angstroms away from the ligand binding pocket? So what we did was, from MD simulations, like, the talk yesterday, they were talking about mutual information, Mohit's talk. we do the mutual information calculation. He did with the sequence analysis, right? He looked at co-evolution, mutual information. What we are, or the gene, like co-expressed genes, mutual information. What we look at is mutual information in torsion angle dynamics. So for example, we know all the torsion angle distribution of all these residues in here. Can we calculate the similarity between the torsion angle distribution in residues that are far away in the G protein coupling interface compared to where the ligand binds to the residues in the ligand bind? So what we do is, from the MD simulations, we calculate the similarity in torsion angle distribution for every pair of residues, and we calculate the average and for the torsion. So once you know, you calculate first order and second order entropy, you can calculate the mutual information. And once you have the mutual information for every pair of residue, we take the average and we look at those pairs that show higher than average. Okay, so because we want to avoid the noise in this mutual information. So, and we do this in torsion angle, then it's less noisy than the Cartesian coordinates. So people calculate mutual information in Cartesian coordinates, which is a lot noisier. So now what we found was the residues in the extracellular part showed a high level of torsion angle correlation with residues in the intracellular part in GPCRs. Now we wanted to map all the residues that mediate this correlation. So we used graph theory algorithms to identify the pathway from this patch of residues to this path of residues, and in the pathway, which are the residues involved. So we did this, what we call as Allosteric, and we applied it to many GPCRs for which we had structures in 2014. And this is the pathway mapped by this graph theory algorithm of communication, allosteric communication from the extracellular side in all these. So when we looked at all the amino acids that are involved in this pathway for over multiple GPCRs, we needed to validate whether what we are doing is right? So these are the amino acid positions that are involved in this allosteric communication. And these positions are common across all these GPCRs that we simulated, right? So they are so we correlated with the mutation results that were available in literature at that time. So what I'm trying to say is, what we found was many of the mutations that are not in the ligand binding site and not in the G protein coupling site interface, but they are in between. People didn't know when these mutations in the 90s, when they mutated some residue in between, that it actually affected the activity of the receptor. They didn't understand how. But what we find is these residues that were mutated and found to be constitutively active mutant or uncoupling mutant, they showed that they were in the allosteric communication pathway. What is more interesting is, across diverse set of receptors, these positions are not conserved. So you could have never figured it out if you compared the sequences and looked for conserved residues in this position. But they are in the same structural position. So what I'm trying to say is, this is in helix 6, position number 43. This is always involved in allosteric communication across GPCRs, many GPCRs, but they are not always the same amino acid in this position. And so we believe that the evolution of these GPCRs has occurred around a structural, a three-dimensional structure. It's like civil engineering, right? If you want certain shape for your structure, your building, you're going to put these heavy joints at certain positions. So these positions are more conserved structural positions than the amino acid type in there. So now, can we use this to calculate ligand bias? So there is something called bias signaling. So for example, in opioid receptor, the addiction from opioid comes because the pain killers that they use to treat opioid, opioid drugs are used for as painkillers, right? So these painkillers are agonists to opioid receptor. They activate two signaling pathways. One signaling pathway goes through G protein coupling. The other signaling pathway goes through arrestin. So the agonists that people are using today as opioid painkillers activates both the pathway. This pathway, this signaling pathway in here, when it's activated, it reduces the pain, feeling of pain in your body. But this signaling pathway, when it is activated, it is going to lead to addiction. So this signaling pathway is leading. So you want to design an opioid agonist that will activate only this signaling pathway and not the coupling to the other pathway, right? So these are called biased ligands. So we thought the intrinsic atomic communication strength should tell you something about the ligand bias. So people are trying to design ligand bias in pharma. They don't know how to use the binding site information from the crystal structure to design these bias ligands. So we came up with a computational way of calculating the strength of these allosteric communication to the G protein coupling site versus the beta-arrestin coupling site, and simply took the ratio, and that correlated well with the experimental bias that people have measured for different receptors and different ligands in these. So now we asked, can we map all these amino acids that are involved in this allosteric communication and simply experimentally test after we predict? So that's what we did in collaboration with Stefan. What he does is he uses FRET as well to identify when you make a mutation in the GPCR, does it couple to the G protein or not, using these sensors, breath sensors. So we predicted in angiotensin receptor, which is an important target for modulating blood pressure. And we identified all the amino acids that are involved in this allosteric communication, and they tested it experimentally based on our, and what we found was that they indeed showed change in how these, when we mutated these residues that are not in the agonist here is a peptide, or in the G protein coupling side, these amino acids which are in between showed a big change in the receptor coupling efficiency to the G protein versus arrestin. And we did that same experiments. We predicted for the, what are the amino acids that are involved in the coupling to arrestin, and we tested that experimentally. So what I'm trying to say is that why is this important? Because if you take cancer, for example, and take a piece of the can, I want to finish with this. So when a patient goes in, they take a biopsy, they take the tissue and identify mutations in a certain protein that is causative for cancer, and they treat that particular inhibitor for that protein as a treatment, right? But mutations occur in this protein, cancer-associated protein, and in different positions in the protein structure. They don't know how these mutations affect the function of that protein. If I can tell the pathologist by doing these simulations and developing this workflow that these mutations are important in, or these residue positions are important in function of that protein, then I can tell whether those mutations will reduce the function of the protein or increase the function of the protein. So we want to develop a workflow that would, computational workflow that would help the pathologist annotate disease-associated mutation, the function of disease-associated mutation. So this can go into the clinic directly, and they can say, ha, this is an important mutation that we should go after for treatment, right? So the more information, currently they only have information that these positions are mutated, and many patients have these mutations, so it is a statistically significant mutation, and therefore we should target it. But what we want to provide is whether this is a gain of function or loss of function mutation in that protein, and therefore whether that would be an additional piece of information for the physician to make this decision whether they should go after that target or not. So with that, I want to conclude. Hopefully I showed that multi-scale simulations provide insights into processes that happen at a microsecond time scale. Like I was saying, the frequency is important. You cannot get that from crystal structure alone. And those contribute to the specificity of this protein-protein interaction. And these computational workflow can be used. I mean, I have pharma funding my research for the past 15 years consecutively, because not because of any greatness, it's because they find these computational workflow very useful to design drugs. So they want this. And we are hoping that the dynamic trajectories are useful to predict and test. So I always like to do experiments concurrently just to show whether our methods are working or not. It's better to know that than to mislead ourselves that it is working. So, and we could have never studied the role of complex lipids on protein function. I didn't show you the results on this. How lipids, external lipids in the cell membrane actually modulate the function is also important. And that type of thing can be done only by simulations, because experiments are very hard for these membrane proteins in this complex mixed bilayer. And then, and I think hopefully I convinced you that computational methods are playing an increasingly important role in biology and medicine. So these physics, chemistry, and mathematics are becoming an integral part of biology. And with all the data coming out, I think mining knowledge from the data is a very important component in medicine. So with that, I'd like to thank my collaborators, Shiv, Chris, Stefan, Hero, Madan, and then Henrik and Brian. I did not talk about all these works we do with them. Thank my group people. The people who are, their names are given in blue are the ones who did the work which I presented, and I thank funding and thank you for your attention.
Yeah. Thank you, Professor Vaidehi. This was a very interesting, elaborative talk, and also it is extremely encouraging to see that computational methods are really useful or becoming useful for predictions for diseases. So it will help the fellow computational chemists and encourage them to take up challenging problems.
Sure.
So with that, I would actually like to open the floor for questions. Although it was already open, but I know if anybody, there is already a question in the chat session that I would like to start with. So Urbashi Nakul has asked that, can you emphasize the noise arises due to the consideration of Cartesian system?
Right. So if you take a simple 4-atom model, right? So a small change in the dihedral angle, like let's say you take let's take what do you call ethane, right? CH3. A small change in the dihedral angle causes a larger change in the translation of the atoms, like the hydrogen atoms in the methyl group, right? So what we saw is that if you were to calculate these correlations or the mutual information in the Cartesian coordinates, there was a lot of noise, especially for those larger amino acids like methionine or arginine. So we couldn't separate the noise from the actual mutual information or like actual correlation in the calculated from the mutual information. But when you take a smaller change in the dihedral angle, that was more meaningful because it was less noisy. The distribution itself was not wide, so it was easier and more numerically robust to calculate the correlation.
Okay, thank you. So any other questions?
Yeah, I have this Prabal here. Very nice talk, Vaidehi, very comprehensive.
Thank you.
So this, I have 2-3 questions actually in this, the multi-component lipid and in that where you embed these proteins and then looking at the coming together of PIP2 or PIP3 to the, so there these proteins were modeled at the atomic level, that was also at the Martini level.
So what we did, Prabal, was first we took this protein in different conformational states, because in the cell they are not in one conformational state. They can be dynamic, right? They can be inactive, active, partially active. So we took the protein structures from multiple conformational states and embedded them in the coarse-grained lipid, and then randomly built the asymmetric bilayer. And we did 50 microsecond we took 5 different random arrangements of these coarse-grained lipids, and then we did 50 microseconds on each. And then what we did was to pluck out certain parts. So there were certain parts where 2 proteins have come together and coalesce. We didn't take that part. We omitted those, and we took those that were monomers because we wanted to look at the monomer function. So we took the protein with its environment, cell membrane environment, and made it all atom, right? But we took out, after we converted to all atom, we took out the protein structure from the coarse grain and threw it and reinserted the crystal structure in the atomic level. So the membrane was coarse grain to all atom converted, but the protein was there in the coarse grain simulation, but we didn't convert that to all atom because that would have lost all the helicity and all in the coarse grain level. So it's not good. The protein structures are not so great when we convert from coarse grain to all atom. So we reinserted the crystal structures.
I see.
That,
but then this another issue, which is like when you have such a multi-component membrane, as you rightly say that the initial, so you can take from a ratio whatever you mention, but then how exactly they actually evolve and the domain formation happens, because that domain formation will critically influence the protein aggregation or the binding of some other components. So that, how do you ensure that in a multi-component membrane, the domain formation, what will be expected in, say, in a real cell membrane versus in this simulation? Is there any control way of doing this, or how?
Yes. So what we calculate, there are multiple things, that properties that you can calculate from the coarse grain to show that you equilibrated the cell membrane well enough. One is we calculate the area of the, it's called the exposed area of the lipid heads, right? And they should be equal for top and bottom layer. That is one way to say that's equilibrated. So we also plot the RDFs, the radial distribution function of all these near the protein of all the different lipids. We know that there are certain markers. Experimentally, we know GM3 is a marker.
Oops, we lost her.
Yeah,
network problem. Yeah,
network problem. I think she's stuck for the moment.
Arnav, you can hear me, correct?
Yes. I was also wondering whether it is my network connection, but I can hear you, so I know that
it's from her end. I see.
Yeah. Okay, we'll wait for a few minutes.
Sure. There is no other question I see in the chat box, or if there are other, I don't know.
No, maybe anybody can ask question directly.
Correct. So I will come back. I have found some more question, but I'll let other.
Can you hear me?
Now she's back now. Yeah. Yes.
So what was I saying? Yeah, we calculate the radial distribution function for different protein for the head groups of the lipids for different lipids, and we know that there are certain lipids that are markers of lipid rafts. For example, GM3 is a marker of lipid raft. See that in our don't know whether that's going to be so we take 5 different equilibrated structures, not just one, and then we do 5 different simulations.
Okay.
If 5 is an arbitrary number, you can do more to be even better. So
I have some more question, but I'll come back after maybe some other people, then I'll come back again. Yeah.
Okay.
Yeah, that's good. So anybody wants to ask question, they can directly ask her or write in the chat box.
All right. I would like to, this is Ambarish. I would like to ask a question.
Yes.
So how do you insert the protein of your interest into this coarse-grained model of lipid membrane?
Okay. So we have a procedure in the lab. I mean, I think this procedure is there in GROMACS as well. So we calculate the principal axis of your protein, like, the three principal axes of the protein. And normally in this protein, it is along the membrane, right? Because most of the center of mass. So you calculate the moments of inertia and diagonalize the matrix, you get the three principal moments, right? So, and we take the highest principal moment, and then you center that in the center of the lipid membrane. There's an automatic protocol, and then you remove the lipids that are clashing. So when I say clash, it is using van der Waals radii. It's less than 75% of the sum of the van der Waals radii between two atoms. Then you remove those lipids, and then you re-equilibrate the lipid in the all atom from the coarse grain. So this is a procedure that we use. If you want, we can share a script, and it is there available in CHARM, GUI, as well as GROMACS. Both have this how to insert the lipid membrane protein into the lipid bilayer. Yeah.
And I guess after inserting the protein, you also equilibrate the system for some time, right?
Yeah, definitely. We do the equilibration for about 70 nanoseconds. So first, I mean, we keep the protein restrained position and distance and also distance restrained between the protein and the ligand, and then you allow only the lipid to equilibrate, the water to equilibrate first, and then the lipid, and then you slowly release the 5 kilocalories to 1 kilocalorie. Slowly release the restrained position restraints on the proteins, and then you do unrestrained for 10 nanoseconds, and then you do the production run. You spawn 5 or 10 velocities on the one starting structure, and then do the simulations, and you aggregate the trajectories, do the analysis. We always do more than one simulation because it's faster that way. You can do it on multiple computers, right, rather than one long simulation. And also with different velocities. So.
Thank you.
Sure.
Yeah, Arnav, before you start, I'll just record my thanks for Vaidehi for giving an excellent talk.